technology. In the previous lecture, I already described the aerobic and anaerobic digestion processes, the biochemical process energy processes of the biomass energy. Today, to describe the alcoholic fermentation process and the biophotolysis process of the biochemical process to produce the biofuel energy. In the alcoholic fermentation process, now I want to describe the alcohol production method. The ethanol C2H5OH is produced naturally by certain microorganisms from sugars under the acidic conditions where the pH is 4 to 5. This alcoholic fermentation process is used worldwide to produce the alcoholic drinks. The most common microorganisms, the yeast Saccharomyces cerevisiae, is poisoned by ethanol C2H5OH concentration greater than 10%, and so stronger concentrations up to 95% are produced by distilling and fractionating as shown in the figure. Hydrolysis and the crush and the yeast fermentation of the 10% ethanol, then distillation process will occur, then we will get the 95% ethanol, then the purification occur to anhydrous alcohol, and which is the petroleum additive or the substitute. When distilled, the remaining constant boiling point mixture is 95% ethanol and the 5% water, which is shown in the figure. The anhydrous ethanol is produced commercially with azeotropic removal of the water by an extra process such as co-distillation with solvents such as benzene or the use of molecular sieves. Only about 0.5% of the energy potential of the sugars is lost during the fermentation, but significant amounts of process heat are required for the concentration and separation processes. This process heat may be provided from the combustion or gasification of otherwise waste biomass and from waste heat recovery. The sugars may be obtained by the following routes: directly from sugar cane, from sugar beet, from sugar cane, from sugar beet, from the starch crops or grains or roots, and the cellulose. Now come to the directly sugar cane. In most the cane producing countries, commercial sucrose is removed from the cane juices, and the remaining molasses used for the alcohol production process as shown in the figure. These molasses themselves have about 55% of sugar content, but if the molasses have the little commercial value, then ethanol production from molasses has favorable commercial possibilities, especially if the cane residue, that is called the baggage, is available to produce the process heat. The major reaction is the conversion of sucrose to ethanol, that is C6H12O6 to 2C2H5OH plus 2CO2. In practice, the yield is limited by other reactions and the increase in mass of yeast. Commercial yields are about 80% of which predicted of the above equation. The fermentation reactions for other sugars, just like glucose or the sucrose C6H12O6 are very similar. The biochemical process of fermentation of sucrose. The chemical equations below summarize the fermentation of sucrose into the ethanol. The alcoholic fermentation converts 1 mole of glucose into the 2 moles of ethanol and 2 moles of carbon dioxide, producing 2 moles of ATP in the process. ATP means adenosine triphosphate. The overall chemical formula for alcoholic fermentation is C6H12O6 to the 2C2H5OH, that is called the ethanol, plus 2 CO2. The sucrose is a sugar composed of a glucose linked to a fructose. In the first step of the alcoholic fermentation, the enzyme invertase cleaves the glycosidic linkage between the glucose and the fructose molecules. The C12H22O11 plus H2O plus invertase to the 2C6H12O6. Next, each glucose molecule is broken down into the 2 pyruvate molecules in a process known as the glycolysis. The glycolysis is summarized by the equation like this: the C6H12O6 plus 2 ADP plus 2 Pi. 2 Pi means the pyruvate inorganic phosphate. Pi is the inorganic phosphate, plus 2 NAD. NAD means nicotinamide adenine dinucleotide ion. Then we will get the 2CH3COCOO minus, which is called the pyruvate, 2 unit pyruvate, plus 2 ATP plus 2 NADH plus 2 H2O plus 2 CO2H. So CH3COCOO minus is called the pyruvate, NADH is called the nicotinamide adenine dinucleotide hydrate, and the Pi is the inorganic phosphate. Now finally the pyruvate is converted to the ethanol and CO2 in 2 steps, regenerating oxidized NAD plus needed for the glycolysis, catalyzed by the pyruvate dicarboxylase. First step is the pyruvate CH3COCOOH plus H plus is CH3CHO plus CO2. Then the CH3CO2 react with the NADH plus hydroxyl ion, that is, then we will get the ethanol C2H5OH plus NAD plus. So from first step the NAD plus to NADH, then again the NADH to the NAD. This cycle is going on. This reaction is catalyzed by the alcohol dehydrogenase, that is called ADH1 in the Baker's yeast. The glycolysis causes the reduction of the 2 molecules of NAD plus to NADH. 2 ADP molecules, adenosine diphosphate, are also converted to the 2 ATP, adenosine triphosphate, and 2 water molecules via the substrate level phosphorylation. Then come to the second source of the sugars, that is the sugar beet. The sugar beet is a mild latitude root crop for obtaining the major supplies of the sugar. The sugar can be fermented, but obtaining process heat from the crop residues is in practice not as straightforward as with the cane sugar. So the ethanol production is more expensive. Third one is the starch corps. The starch corps means the grain and the cassava, which is shown in the figure, can be hydrolyzed to the sugars through the hydrolysis. Starch is the main energy storage carbohydrate of the plants. and is composed of two large molecular weight components. One is the amylose and other one is the amylopectin. These relatively large molecules are essentially linear but have branched chains of the glucose molecules linked by the distinctive carbon bonds. These links can be broken by the enzymes from the malts associated with the specific crops, such that the barley or the corn, or by the enzymes from the certain molds, like fungi. The links can also be broken by the acid treatment at the pH is 1.5 and the 2 atmosphere pressure, but yields are small and the process more expensive than the enzyme alternatives. An important byproduct of the enzyme process is the residue used for the cattle feed or the soil conditioning. Number 4 is very much important, that is the cellulose. Wood or the plant stalks. The cellulose comprises about 40% of the all biomass dry matter, including the crop residues remaining after the grains, juices and the fruits have been removed. Previously we have noted its important use as a combustion fuel, but it also has the potential to be a major material for the ethanol production. The ethanol from the cellulose is therefore often called the second generation of the bioethanol. The cellulose, which have the molecular weight is 5 into 10 to the power 5, has a polymer structure of the linked glucose molecules and forms the main mechanical structure component of the woody parts of the plant. These links are considerably more resistant to break down into the sugars under the hydrolysis. This is also the hydrolysis process than the equivalent link in starch. In plants, the cellulose is found in the close association with the 15 to 25% by mass of the lignin, a polymer which is even harder to break down than cellulose. Thus, these woody feedstocks, including the grasses and the stalks, are collectively called the lignocellulose. Now the second part of the biofuel production, that is the biophotolysis process. The photoautotropic organisms such as microalgae and the cyanobacteria are capable to generate the hydrogen and the carbon dioxide in the biophotolysis process. This is the bacterial photosystem. The input is the sunlight and the organic acid. Then the ferredoxin is the catalyst and the ATP is also the catalyst. Then we will get the energy source and the carbon dioxide as carbon source to split the water into the hydrogen by utilizing the light. They produce the biohydrogen by the direct photolysis of the water under the anaerobic conditions. There are two types of biophotolysis, which are direct biophotolysis and the indirect biophotolysis. In the direct biophotolysis, the hydrogen production by direct biophotolysis process is used the solar energy and the photosynthetic system of the algae to convert the water into the chemical energy, which is subdivided into the one stage or the two stages. In direct biophotolysis, the light is absorbed by the photosystem 1. This is the PS1, and the photosystem 2, that is PS2, which use in hydrogen production and energy to the transport electrons linearly to ferredoxin. The light absorbed by the PS2 is required for the oxidation of the water into the electrons, protons and the oxygen molecules. These electrons then transfer through the electron transport chain using the light energy absorbed in PS1 to the ferredoxin to the hydrogenase enzyme without involving the intermediates of the carbon dioxide fixation. These enzymes are then catalysts for the recombination process of the proton and electrons to evolve the hydrogen gas. This mechanism also refers as the one-stage direct photolysis, where the biohydrogen is produced directly from the water and the solar energy via the algae photosynthesis system. The equation likes this, that the 2H2O is converted to the hydroxyl ion and the electron and the oxygen. Then the 4 hydrogen ion plus 4 electron is equal to 2H2, the hydrogen generation. There is a major limitation for this process, which is the sensitivity of the FeF hydrogenase enzyme to oxygen that produced by the photosynthesis, which greatly prohibit the activity of the enzyme for evaluation of the molecular hydrogen and resulting low yield of biohydrogen production. Meanwhile, the two-stage direct biophotolysis is a continuous and upgrade process from one-stage direct biophotolysis, which the oxygen is removed through the respiration using the exogenous or the endogenous substrates such as sulfur deprivation method. Now come to the indirect biophotolysis method. The indirect biophotolysis is the production of hydrogen from the water via the microalgae and the cyanobacteria photosynthetic system to convert the solar energy into the chemical energy, which is hydrogen, by two steps in series. The first step is the biomass production through the photosynthetic system, and followed by the second step, which utilize the biomass rich carbohydrate for the hydrogen producing fermentation. First step is the 6H2O plus 6CO2 plus light. Then we will get the C6H12O6 plus 6O2. Then the C6H12O6 plus 2H2O, we will get the 4H2, the acetic acid, CH3COOH and the carbon dioxide. In this process, the evolutions of the hydrogen and the oxygen are temporarily and or spatially separation at different steps. In this way, the inhibition of the hydrogenase enzyme activity to oxygen produced during the photosynthesis process can be avoided. The examples of the cyanobacteria organism that perform indirect photolysis, such as Globibacter species, Sinosystis species or the Psychococcus species. The advantage of the biophotolysis is the simplicity of the process to produce the hydrogen without requirement of the additional substrates as the nutrient, and can produce the hydrogen directly from the abundant source of the water as electron donor, sunlight and the carbon dioxide as the basic needed for the growth of the microalgae and the cyanobacteria. However, the process is required the high light intensity, and the green algae and the cyanobacteria have the lower light conversion efficiency. The lower performance of the photobioreactors of the light penetration in the dense culture is contributed to the lower hydrogen production. These are the references from where I make this lecture. You should go through these references, then you will get some more knowledge. Thank you.